recognize the virus, because it turns out most people with chronic hepatitis B are not effectively recognizing the virus as foreign. So maybe there are ways we can teach the immune system to do that. So 13 years ago, to start with on our drug watch, there were about 8 or 9 drugs on our drug watch, and I've divided them into the direct-acting antivirals and then the indirect-acting antivirals at the bottom. A few years later, in 2010, there were much more, and I only go to 2015. I loaded the wrong one, sorry. I had the 2019 one loaded. There are now more than 31 drugs in the pipeline, and now you can see why we're very excited at the Hepatitis B Foundation, because of the future, we think, in front of us for treating hepatitis B. These are preclinical drugs over here. They're still being tested in the laboratory and in animals, and these are already in the human phase, what are called clinical phase 1, 2, or 3 drugs. And of course, you know, Benlydia was approved about 2 years ago, the newest hepatitis B drug to be approved, another polymerase inhibitor. But look at all the things happening out there. These are direct-acting antivirals in blue, and then the indirects in gray. Now let's walk through them step by step, starting with the beginning of infection, and give a sense of how they're working. And then I'll show you lab value charts from people treated with these drugs. So we're only really going to talk about the clinical phase drugs, the ones under here, the dark blue. Well, this is entry, up in the upper left-hand corner, drugs that prevent entry. I would have never thought these drugs were going to be any good, but actually they've turned out to be very impressive. Merclid XB is a drug that blocks entry. I'm showing you entry here. Here's the green virus attaching to the cell. It prevents that from happening. Contravir, we are on the board of, for full disclosure, has a drug that also does that. But the Merclid XB drug is well advanced into phase 3, very close to being approved for use by the FDA. Now, it's been tested mostly in people with hepatitis D as well as B. That's a complication that I think you'll hear about later. But the results have been nothing short of astounding. What you're seeing here is, of course, the D level drop. So again, this is one slice of the chart I showed you. This is down here on if you follow me here. This is time. Now it's only 48 weeks, and this is the level going up and down of the analyte, whether it's DNA here going down, or in this case, look at this. Walk over here so you see. This is surface antigen. We don't see that in most of the drugs that we're using. Surface antigen is dropping. That's what we really want to see. And but not that. What happened to the? That was bad. Something. All right. Hey, what happened? The slide thing. All right. Well, the lights. Did the lights dim? Well, anyway. That's. Did I hit something?
I don't think so. We got it.
All right. Well. Thanks. All right. Well, now I don't even notice when the lights on.
Just push that button. There you go.
All right. So, so that's Merclid XB. The next on the list would be next on the list would be a drug that directly attacks surface antigen, and maybe I'll do a little bit of boasting. So surface antigen is the empty particle that's in your bloodstream. There are drugs out there now. That will target surface antigen, and some of those I want to say originated at the Blumberg Institute of the Hepatitis B Foundation. We were among the first to actually be screened for those drugs. Now, our drugs haven't made it to the clinic, but that's the kind of work we do. Probably the drug that is most advanced, and I wish you could see the slide of it, is a drug called Replicor, or from a company called Replicor, and that is what is called a nucleic acid polymer. No one really knows how it works, but in individuals, and it's an injectable, so I don't like that. In people treated with this Replicor product, also treated with interferon, also treated with vaccine, so people are on multiple drugs. That is a drug that was able to reduce S antigen in 8 out of the 12 people on the slide I had. Now they've repeated it, and in 6 out of those 8 people, antibodies appeared for hepatitis B. That's a definition of a resolved infection. So we're very excited about that. And what I want to tell you is that drug, and people are on the edge of their seats for this, is now going to be tested. Oh, thanks. Is now going to be tested. What happened? Was it me? Was it me? All right, I'm going to use this instead. All right, so I'll advance the Replicor slide. So we were here. I'll go back to the word. Oh, this is Replicor, and then I'm going to have to go back and forward because I got. So this was Replicor, and here these individuals are. So Replicor is treating this suboral particle, and S antigen levels drop. That's what you're seeing here. This red line drops to below detection. DNA levels drop because the people are on tenofovir, so that's no big surprise. They're also vaccinated. But look here, red coming up. This is antibody. That's the best. That's the best. That's beyond really. That's even beyond the endpoints that are predicted, are expected for most of the drugs out there. Now let me go back and tell you about another category of drugs called the capsid inhibitor. So you'll recall I mentioned that the hepatitis B virus, if you look in this cartoon on the upper left-hand side again, you can't really see it. You can't see my pointer, but you see those red lines, dashed red lines, are showing that the capsid forms from monomers, single monomers. Oh, that's all right. I'm afraid to make the slide go away. Okay. Okay. So a capsid forms from right here, and then that normally that forms the nucleocapsid. There are now drugs out there that prevent that from happening. They cause the particles to be either empty or misformed and never touch RNA or DNA. And I again want to point out, not to be boastful, that some of the initial capsid inhibitors came from the Blumberg Institute. And actually our capsid inhibitor design is the foundational design or patent that all the others that are derived from, that are in clinical use. Ours probably won't see approval. They're under development by Arbutus, but drugs just like it are. And they're having effects that people expect. Here you again see that these are people treated with capsid inhibitors, and you see the viral DNA levels drop, and I believe these are the assembly molecules, but you see the same results from Arbutus. The other companies all have drugs out there that are inhibiting capsid, and this is the effect they're having. We wanted to see more, to be honest. We were hoping to see surface antigen drop as well. We haven't seen that yet. That may yet come from combination trials that we'll talk about. Now, there's a family of drugs that I'm really very excited about called the RNA Degrading Molecules. And I'm going to run out of time, but I think I should have some time added because of this. Called RNA Degrading Molecules. And what you'll see here is these are molecules initially identified and reported by Roche, and then brought on to Arbutus by actually Chris Moore, who's in the audience. And I think they're very clever. We're working with on them to figure out how these things really work and get new ones. But they cause hepatitis B virus RNA. This black stuff here is hepatitis B RNA from an infected cell in culture. And all that RNA just disappears. And of course, the viral DNA levels then, of course, disappear. These are animal studies, not yet people studies. I put it in here because of the role of Blumberg, of course. And I think these are some of the most inventive and creative drugs out there. And I hope that they'll be in clinical trials within the next year. What is in very advanced stages in clinical trials are what are called RNAi molecules. These are gene silencing type molecules, and they actually attack hepatitis B virus RNA after it's made and cause it to be destroyed, kind of like the chemical I just told you about, the RNA degrading molecule. But these guys are already in clinical trials. Look here in the lower right-hand side here. I have to figure out how to do this. You see that viral DNA levels drop from this one human study, that's Arbutus even. And this is given to me from the Arrowhead study. And well, again, what you see here is S antigen. Look at the upper left-hand panel. S antigen is dropping these individuals. That's just what you want to see. That's very exciting. And actually, when put in combination with a vaccine, this was an animal study though, what you can see is a liver resolution, histological resolution, and also antibody. Oh, I didn't show that here. Where's the antibody?
It's at the bottom.
Oh, I'm sorry.
Bottom left.
All right, bottom left. Oh, yeah, antibody coming up. Again, the perfect trifecta: DNA gone, S antigen gone, and antibody coming up. So that's what you want to see. cccDNA is, of course, the dreaded parent of all hepatitis B virus products. People are trying to target that. No drugs yet. Again, the Hepatitis B Foundation's Blumberg Institute was the first to develop assays for cccDNA and the first to introduce a drug that targets cccDNA. Of course, it didn't survive. It didn't survive development, but I like to think that it was a pioneer and moved the field forward. You know, as Dr. Cohen pointed out, Hepatitis B Foundation actually with 34 other thought leaders, individuals in the country, generated a roadmap for a cure research agenda, which the NIH is actually using today to National Institutes of Health to identify projects that needed to be done to find a cure, and we'll be convening that group again this fall. So the Hepatitis B Foundation and Blumberg Institute, I want you to know, has a hand. It has in almost every important initiative involving hepatitis B cure. Immune modulators. Now I'm going to go through this in just 2 minutes and then invite Chris Moore to come up and talk about it, because he's an immunologist and could probably do a better job of this. But all I want you to see here is look at the number of drugs now that are in clinical trials and in preclinical that are targeting the immune system. I've just told you about direct-acting antivirals. This is targeting the immune system. This is a table from Jiangmi Chen's paper that came out a couple of years ago, just summarizing the clinical trials going on around the world, but registered with the NIH in the area of immunotherapy. And immunotherapy, again, is the different side of the cell. So here's the infected cell. I was talking about the virus. These are the immune cells that we need to educate to attack our infected cells, and then we want and also to produce antibodies. In people who have hepatitis B right now, the problem is it appears that immune cells are tolerized. They're turned off. They're told to not recognize hepatitis B proteins, and hence they don't effectively target the cells. They do just enough to cause trouble, but not enough to clear the virus. I'm not going to go through, there are a number of clinical trials. They have all been disappointing so far for immunotherapy for hepatitis B. They show that they work biochemically. They have not worked clinically to the benefit, except for one category that I'm going to run to, and those are what are called the program death 1 regulators. So this is an all-new area. It's very exciting in cancer therapy. We have on our cells proteins that recognize that are told to be turned off, that turn off the T cells. I said our T cells in Hep B are turned off. Well, now the proteins that are turning them off have been identified, at least some of them, and there are drugs out there now that reverse that, called PD-1 inhibitors or PD-L1 agonists, and they'll work on either sides of this. They're used in cancer, and they're showing remarkable results in cancer, and some scientists are using them to treat hepatitis B. And what I'll show you here is a study of novelimab, which is an antibody that works on that PD-1 receptor. And each bar here is a different person showing the level of HBsAg 12 weeks and 24 weeks after treatment with such a drug. And you'll see almost everybody in this study experienced reductions in S antigen. Some pretty significant, most on the borderline, but showing real promise. So I think that the future is going to be a combination of therapy, and I think people will continue to take direct-acting antivirals, maybe a capsid inhibitor, maybe an RNA degrading inhibitor, certainly a polymerase inhibitor, and ideally that will be taken with an immunomodulator to induce this kind of double punch treatment of hepatitis B. And we're going to see, look, we're way down here now. How many people get cured from being treated for hepatitis B now? About 5% with the current drugs that are out there. People do get cured. A long-term treatment, tenofovir, what we call a virological cure. Just like with hepatitis C virus, after this first round of new drugs, it was about 10%, then it was 20%, then 50%, and then finally up to 95 to 100% of people were cured for hepatitis C. We're going to get there. I feel quite confident about that, but it's going to take time and patience. So I'm going to thank the people who gave me slides and helped me with this. They're on the list, and I thank them. I'll take a couple questions, and then I'll ask Chris Moore to say a few things. Thank you. Are there any questions? If not, Chris, do you want to say anything? And so, Chris, and also can talk about some patient outreach.
Come up there?
Yeah. So you can be here. Is that all right? Thank you. All right, we're running late, so just a few minutes. We're running late, not but
just a minute.
It's
but
not,
you know,
yeah, I know.
not my fault.
It's not your fault, yeah.
Hello, yeah, my name is Chris Moore. I'm the head of immunology at Arbutus Biopharma. Yeah, I think that Tim framed out the potential therapeutics that are out there quite well. A couple things I just wanted to add that we think about, because we actually, we work on this every single day. This is what we do for a living, is try to make a cure for HBV. I was fortunate enough to work in HCV for about a decade, and I was around when that cure came out. And in fact, I came up here to Pennsylvania from North Carolina to work for the man that invented sofosbuvir, which became Harboni, the cure for hepatitis C, Mike Sophia, who's also a faculty member at the Bloomberg. And so we switched to work on HBV and try to cure that infection. Now, HBV is a much more interesting virus, and when a scientist says interesting, that typically means that it's very hard, very difficult to cure. And one of the reasons for HBV that Tim was talking about is that there is this immunological component to HBV. The problem with HBV that's different from HCV is that it pumps out literally almost teaspoon amounts of protein called HBs antigen. And that S antigen floats around in your serum, and eventually your immune system looks at that S antigen and says, I'm not going to recognize this anymore. And so essentially, HBV has the ability to turn off the immune system to itself, and your body will see HBV as self. So that's the definition of tolerance. And that's what's happening in HBV. Now, luckily, in HBV, there are patients that just spontaneously resolve HBV. So it's about 1% on nucleoside therapy. And in those patients, their own immune system recovered, reawakened, and it saw HBV as foreign, and it cleared it. And now those patients are cured, and they're essentially like a vaccinated patient at that point. And they have antibodies floating around that's able to protect them from future infections. So what I'm trying to do with my group is we're trying to reawaken that immune system to HBV. I think the key in HBV will be immunotherapy. Tim mentioned the capsid inhibitors and nuke therapies. We're making a lot of progress in terms of suppressing the virus. The virus now, with the capsid plus a nuke therapy, which was the clinical trial that was just completed, essentially goes to undetectable. But those patients still don't cure. At least they didn't cure in 24-week therapy with the combo study. So probably you're going to need to boost the immune system. The problem with boosting the immune system, as you know, is safety. And for HBV patients, safety is paramount because these patients typically feel okay. And a lot of the therapies that we want to use as an immunotherapy are similar. In fact, the same mechanism of action as some of the immunotherapies that are used in cancer patients. For example, Opdivo or Keytruda that you may have heard of is anti-PD-1 antibodies that are immunotherapies. Those come with significant risk and have a lot of adverse events. And And the one study that I will point to that Tim showed was the study by Gilead, where they took Opdivo, or actually I think it was Keytruda, and they tested it in patients. They did not do anything with S antigen. That patient still had lots and lots of S antigen. They gave a very small, low dose injection of Opdivo, tenfold less than they would give a cancer patient. They saw no adverse events in those patients, and 1 out of 12 of the patients totally cured. And so that gives us a lot of hope that with suppressing the virus and then coming in with something to boost the immune system, that we will take that cure rate from the 1 to 3% that it is now to upwards of 30 to 40 to 50% over the next few years, hopefully the next couple years. So that's all I have to add here.
Thank you, Chris. Thanks for explaining that. So we are going to, I'm going to make, hold on. I can't stay where I am.
Oh, Sue Wong at the top. Sue
Thank
Wong.
you.
There
I can't see. All right, so we're going to take a step back. We were just talking about, cells and immunology. Now we're going to take a really big step back and talk about the world and policy and stuff that is impacts the lives of so many people. And this is, and I'm going to make it short because we're running late. But I'm really glad to be here. You turn that on. Hello, is this better? Thank you, Martha. So I am the medical director for Center for Asian Health. I used to work in New York City, in Chinatown, where there were a lot of Asian patients, and so I became very familiar with taking care of Hep B patients from a primary care perspective. And I have, over time, also began to really recognize the importance of the patient voice and how important it is, not just physicians, not just policymakers. And as a patient myself of Hep B, I've also come to realize that this is one of my most important roles in my career, is to actually represent the patient voice, not just the physician voice. And so I have become really involved with HBV Foundation, also the World Hepatitis Alliance, which represents patients on a global level. And so I think this is a super exciting time for patients to be engaged with what's happening in the scientific and research realm. So I think hopefully, some of the basics everybody knows. The most common infection in the world, liver cancer on the right, liver cancer rates are on the rise. So we know whatever we're doing now is not working. This is globally the distribution. And so the US looks like, oh, it's not a big deal, but the problem is you're not looking at the targeted populations. And so when you look within the Asian populations, the African populations within the US, you see really, really alarmingly high rates of Hep B. But what's exciting is we are currently in the era of elimination because we have all the tools. We have diagnostics, we have vaccines, we have tests, we have treatment. For Hep C, we have cure. For Hep B, we're hopeful for the cure as well. And so in 2016, the WHO basically said, we believe we can eliminate hepatitis B and C by 2030. So how exciting is that to be able to pass that on to our children, our legacy, to say, you know what, we remember when there was Hep B and Hep C, and it's no more, kind of like polio. And smallpox as well. And measles, we thought, until people stopped vaccinating. So, and then in the US followed in 2018, the Institute of Medicine Convened a committee and said, we can do it in the US, but it does require significant resource allocation, commitment, and strategy. So this is kind of shows you where we are in the race to eliminate, and there are multiple steps. We still have a lot of work to do with screening. And I think this is one thing we in the advocacy world always try to say, hey, this actually has a lot to do with policy and advocacy, because hepatitis is underfunded, even though it impacts way more people than HIV. You can look at the numbers here for Hep B and Hep C, and Hep is 2.2 million and almost 3 million compared to HIV million. Yet when you look at the division of the money, most of it, 788 million just for HIV versus 39 million. And what does that mean? So that means, it impacts research, programs, surveillance, public awareness, provider awareness, and general progress for this. And so I think it's important for us to realize that a lot of this money actually has to do with Congress, and Congress is often listening to its constituents. And we think Congress will just look at the numbers and make good decisions, but they don't. They often will listen to patients and individuals, and it's the stories, and that's why the Just Be campaign is so important, because as Hep B patients, we really need to be willing to tell our stories, because that's what often is what changes people's minds and helps them decide what they believe is, is important. So, and we know that unfortunately, even though we were making progress, Hep B and Hep C are both on the rise largely because of the opioid crisis. And so we need to be innovative. What are the things we need? We need to scale up screening. We need more access. We need frontline workers, primary care doctors like me to do more with screening and treatment. We need access to medications. We need the guidelines to be easier to follow, and vaccines, not just for kids, but also for adults. And we need a movement of patients' community, and that's really my big message for today. And all of this is like a big synergy with research as well. So the World Hepatitis Alliance, we're doing a 3-year initiative called Find the Missing Millions. So you'll see this. We have, if you go to our website and the No Hep campaign, if all of you can join, that will actually make a big impact too. Go on the website, join for No Hep. And all these resources are available for people to use. Those of you who use social media, July 28th, this is a great chance for everybody to raise their voice and say hepatitis B is important. Tweet these things, and you young people can use Instagram and Snapchat or whatever. Because globally we're still failing and not doing a great job with hepatitis B and C diagnosis, but definitely worse with hepatitis B. So it's been recommended, people always say, and I get this from doctors too, I don't want my patient to have to pay for it, it's not covered, and they'll actually, I feel like, scare their patients by saying that, and so they won't order the test. But it's been recommended for a number of years that under ACA, that high-risk populations, which includes all of us Asians, should be screened for hepatitis B, and it is covered by Medicare as well. All right, I'm going to skip this. And every day we see patients who say, how come nobody screened me? Somebody with cancer who reactivated. And that's what's different about people who have had Hep B in the past. If your core is positive, is that the virus is very smart and actually reactivate, and it's devastating when it happens. People who've been to the doctor a million times, they have stroke, they have diabetes, but nobody bothered to check their Hep B. That's somebody we found in the emergency room. We have an emergency room program where, what I said, we have to be innovative. So besides just you going to the doctor to get tested, we got to think of other ways to screen people. And so in our emergency room, we started doing an automatic algorithm. We started collecting country of birth, and it automatically orders a blood test if somebody comes from that country, a country that's considered endemic for Hep B. And then we have navigator, my navigators, wave your hands, who are awesome at getting people who, oftentimes the emergency room is where people go when they don't have care. And so they're really helpful with that individualized attention to get them to the doctor. And people will just say, you know what, I just didn't feel, I wasn't motivated, I lost hope, I didn't think the cure was for me or for the Hep B test was for me, and having somebody just advocate for you and say, I'll make the appointment for you, I'll meet you there, makes all the difference. So you can see, obviously, when you automate something, our numbers went up. And this is just really interesting. This has been education to us, because traditionally we have done only outreach in the Asian population and African also. But when we did this Hep B screen, we were surprised that we had so many Haitian patients in our community. And that they end up being a community that has Hep B. A lot of Caribbeans, as you see here, Jamaica, Dominican Republic, Africans. And so it's just a message that it's an international, global disease. It's not, doesn't have one face to it. And so we've learned a lot from doing this program as well. You can see the prevalence rates go all the way up to like 13% for Guinea, and Liberia like 10%. So mother-to-child transmission, if we can eliminate perinatal Hep B, that is a key part of really doing elimination. We are still getting babies infected with Hep B, and a lot of it is because babies aren't getting birth dose. So this is something that we're trying to push advocacy-wise too. And people say, oh, we're okay at 70%, but it's a shame when you look at other countries in the Americas, like Mexico's 90%, Cuba's 99%, so it's not just a matter of resources, it's a will, it's a commitment issue. We don't prioritize it in the US. And this just shows modeling in terms of if we want to get to elimination by 2030, what are the big impacts for maternal to child transmission? And you can see the big jump from up here to down here is that birth dose. I mean, the babies need to get it within 24 hours, not wait until they go see the doctor at a week. So we need a movement. We need all you guys part of it. It's not just about science, it's really about a movement. This is a screening we did in our hospital. I think no matter what you do or where you are, you can make a difference in your circle. This was at a medical conference where we actually had our first ever kind of community exhibit. It was so different from like a lot of the medical conferences. It was very vibrant. We had this TP here. So it's great when the community comes out. You see this at the HIV conferences. It's a complete celebration of community, and we need to see that too for Hep B. And what we say is, you know, we may not have a lot of money in Hep B, but we have a whole heck of a lot of people who have Hep B, and so let's utilize what we do have, which is a lot of people with Hep B, and they, you know, we as patients in the community can drive the desire for, you know, awareness and want prevention. We need to demand access and encourage innovation so that we can all move forward for Hep B. So I would say, you know what, what's great is we're starting to see more public media awareness, and you know, I was really excited to be featured in the Science article, which HBV Foundation was featured in too. And so it's hitting the mainstream now, I feel like, finally. We just need to keep that going. The Lancet actually had a big article about hepatitis elimination, and this is a great article in Nature too. So these are good articles to read that are really less, you know, just for the scientific community, but more for the wider community. So this just shows step by step onto 2030, which is really only 11 years away. You know, we got to make some progress to hit elimination. So thank you all. We are going to go into our breakouts. So everybody should have the schedule of different workshops we have. So we're all going to take a step out, you know, get your wake up, and then they're going to turn the room around. All right. So there's pizza. And there's pizza. You know, we ran out of food. Please, pizza break.
Grab pizza. We're going to break the room into two and come back in a few minutes.
Don't leave, please. The workshops are going to be really excellent. It's a chance to really interact. You know, I think it's hard to be in a lecture